Your question or your we'll call it a problem. Please decode mRNA strand ACC, please. Please decode the mRNA strand ACC, please. Type in the amino acid that it codes for. Please decode ACC. Bless you. All right, let's see what you guys put. Should be pretty straightforward. I'll lock you out. See what you have. Threonine is right. Threonine is right. Threonine. No, not tryptophan. I said mRNA is ACC. We'll talk about that in a minute. I didn't ask you for UGG. So I gave you the code ACC. I said the mRNA is ACC. If the mRNA is ACC, the first one is A, the second one is C, the third one is C, so there's ACC, right? So it's threonine. You did the anticodon. If you do the anticodon, you get like tryptophan, right? That's wrong. So if you don't use the mRNA codon and use the anticodon, are you going to get the right amino acid? No, that's why it's called a codon chart, right? Because you use the codon. It's not called an anticodon chart. It's called a codon chart. Okay? Is this the genetic code? Yeah. Okay. Yeah. Ready? Take a mental image. No, I'll give you the chart. I would give you the chart. What? Sure. Here we go. Let's do a different one this time. Give me a different one. I don't care what it is. AGC, I heard. AGC. Oh, I just wrote A. Well, here. C. You ever have your hand do something your mouth doesn't? All right. A, first one. G, second one. C, third one. So we go here, and the last one is C, which is here. So there's AGC right there, right? So that's serine. We good? 1, 2, 3. 1, 2, 3. All right. So today's more of a review, kind of what we just talked about yesterday, and then I'll let you work. But we got to get through it first. So translation occurs in a sequence of steps. This involves 3 kinds of RNA and results in a complete what's called a polypeptide. Poly means many. Peptide referring to the peptide bonds between the amino acids, right? We copy DNA onto RNA. What type of RNA? mRNA. What's that process called? Transcription. So we copy DNA onto RNA. What type of RNA? mRNA. What's that process called? mRNA wiggles through goes out into to assemble proteins by assembling and forming peptide bonds. What's that process called? Translation. What are the 3-word messages on mRNA? Codons. So ribosomes read 3-letter words called codons, and they match with, everybody say anticodons, anticodons on a, everybody say tRNA molecule, which also carries an amino acid. 2 amino acids next to each other will form a peptide bond. Okay? Yeah, there you go. There you go. Now you're on it. Translation takes place in the cytoplasm. We understand that outside of the nucleus, where tRNA, rRNA, and mRNA all come together. So mRNA comes from the nucleus out into the cytoplasm at the ribosome. tRNA brings its anticodon to match the codon on mRNA. And where's rRNA? That's just what, well, it is, but it is in the ribosomes. Okay, ribosomes are made of rRNA. That's why it's called ribosomal RNA. Okay? Class is too short to be taking people out, whoever you are. Am I too loud? Hold on. Here we go. Too loud? Oh. My neighbor. Oh, the REDI, R-E-D-I experiment. The REDI experiment. Yeah, that was to prove Operon's theory of spontaneous generation. Yeah, yep. How you get, well, you get living organic compounds from non-living inorganic elements. Yep, you got it. It's Operon, O-P-A-R-I-N. But that's his, it was Operon's theory, but it was REDI who did the experiment. Okay, yep, bye. All right, so what? He asked what was the experiment that you can make inorganic or living molecules from inorganic matter. And it was Operon's theory that Thomas REDI actually did the experiment to prove it. So what? The peptide bond is between the amino acids. I'll show you in a second. Hold on. So a specific amino acid is added to one end of each tRNA. How do I know which amino acid? It is off of the codon chart, right? So we use the codons. Where are the codons? They are on, everybody say the code is on mRNA. We copy DNA onto our mRNA most specifically. Let's just keep it RNA and then we'll change it in a minute. We copy DNA onto RNA that we copy is called what? mRNA. What's the process of copying RNA called? Transcription. What are the three-letter words on mRNA? Codons. So mRNA with its codons wiggles through a nuclear pore, goes out into the cytoplasm to find a ribosome. What are ribosomes made of? Everybody say rRNA. So the mRNA codons are read by matching codons with, everybody say anticodons. Anticodons, which are found on tRNA, which is also carrying in amino acid. If I get two amino acids next to each other, they form a peptide bond. Okay? So an anticodon is a three-letter nucleotide sequence on tRNA that is complementary to mRNA, but do not dare read the tRNA molecule to get your amino acid, or you will have the wrong amino acid. You need to use the codon, which is on mRNA. How do I remember that? Before mRNA leaves the nucleus, DNA says, hey, put your codon, right? Yeah, okay. So the mRNA joins with a ribosome and tRNA. A tRNA molecule that has the correct anticodon and amino acid binds to the second codon in the mRNA strand. I will tell you, you can fit two codons inside of a ribosome at a time. Would I ever need to have more than two inside of there? Would I ever need to fit more than two codons into a ribosome? No, because once two amino acids get next to each other, they form a what? Peptide bond, and I don't have to hold them in place anymore. So a peptide bond will form between the two amino acids, and the first tRNA is released from the ribosome. Now the ribosome then will move one codon down the mRNA strand. This just keeps happening, thousands of amino acids, until we reach what? The stop codon. What is the stop codon code for? Say absolutely nothing. Absolutely nothing. If I put nothing on a chain, the chain is broken, right? And then I have my polypeptide. Okay. We'll talk more about it in a minute. Clicker's not working. There we go. So the amino acid chain continues to grow as each new amino acid binds to the chain and the previous tRNA is then released. Hey, so if my tRNA matches the anticodon, drops off its amino acid, then it goes and finds what? If somebody took my ball, I go look for another ball. If someone takes my amino acid, I go look for another amino acid. So tRNA is going to go out and find another amino acid. Where does it find it at? How do amino acids get into your body? Food, right? What are the monomers? Like, is there a lot of protein in eggs and meat? Yeah, so you break proteins down into their monomers called what? Amino acids. And then you take those amino acids and you make your proteins, right? So if you don't eat, can your tRNA find an amino acid to add onto the chain? No, and then you don't make your proteins, and then you die of starvation, malnutrition. You can't make proteins, you can't grow muscle, you get emaciated. You get the idea, right? So I know we talk about all these different little microscopic parts, but keep in mind, they're building you, right? They're building a structure like a muscle. So this process keeps going until you reach a stop codon, and again, that codes for nothing, so the chain is broken. Many copies of the same protein can be made rapidly from a single mRNA molecule because several ribosomes can translate the same mRNA at the same time. So here's the thing that I don't really talk about. Imagine a long strand of mRNA that goes from here to the other side of the room. Think of it as a rope, right? Imagine we are all ribosomes. Could we all grab the rope at different places? And then we could I could start reading the rope here, and then someone come right behind me, they read the rope, and then someone behind them, and they read the rope. So you have thousands of these ribosomes all reading the same mRNA strand. You make 4 trillion hemoglobin proteins a minute. That's one protein. What does hemoglobin do? It carries what? Sorry. Oxygen, right? Hemoglobin is the protein in your blood that carries oxygen. That's just one protein, 4 trillion a minute. So we must be reading that mRNA strand thousands of times, right? Think about it. Because we got to keep pumping out that hemoglobin because every time I take a breath, I've got oxygen. Something better hold on to it or I die. I don't go through electron transport chain, right? So what does it look like? Oh, so let's put it all together. Inside of the nucleus, stay with me here, we're almost done, I promise, but do not, I repeat, do not zone out right now. So inside of the nucleus, we have this very special double helical structure called DNA, right? Contains all of our 30,000 genes. How many genes do we copy at a time? 1. So RNA is very small. We copy DNA onto RNA. What type of RNA? mRNA. There are 3-letter words on mRNA called codons. What's the process of copying DNA onto RNA called? Transcription. So mRNA with its codons wiggles through a nuclear pore, goes out into the cytoplasm to find a ribosome to make a protein by assembling amino acids. Now, it does this by matching the mRNA codons with anticodons, which are found on the tRNA molecule, which is also carrying an amino acid, right? This process is called translation, okay? Now, if I get 2 tRNA molecules inside of a ribosome next to each other, this is called a P site, this is called an A site. Don't worry about why, it doesn't matter. Now, if they do get next to each other, they'll form a peptide bond, like you told me. And at this point, does tRNA have to hold that amino acid together? No, because it's a peptide bond, right? It'll stay. So this just keeps happening over and over and over and over and over and over again. Do you guys remember the code that starts the whole thing? If you start work, you go UG, right? AUG. Okay, so if you have to start working, you go UG. So it's AUG, which is methionine, right? Okay, so all amino acids start with the same, but they don't all end the same. There are 3 different stop codons. A stop codon will code for absolutely what? Nothing, and the chain will be released. Now, immediately we call that a polypeptide. Everybody say polypeptide. Is it automatically a protein? Do you guys remember primary, secondary, tertiary, and quaternary folding of proteins? Whew, that was a while back, wasn't it? Okay. Back when we were talking about remember how proteins fold? That was a while. It was like chemistry back at the beginning, chapter 2 or something. So polypeptides do not immediately function. If they do function, they're a protein. So say this with me. All proteins are polypeptides, but not all polypeptides are proteins. So all proteins are polypeptides, but not all polypeptides are proteins. Give you an example of that. There's a polypeptide called a prion. It causes mad cow disease, but it's not a protein. Okay? So sometimes proteins are destructive, right? Sometimes polypeptides are destructive too. All right. I don't want you to get confused with that, but really all you have to understand is this is how we make a protein. Okay? A long chain of amino acids is immediately called a polypeptide. If it serves a function, it's then a protein. Okay? So it's not always as easy as just making a polypeptide, right? Sometimes we have to identify how and what gene expression is influenced by. So the relationship between genes and their effects is complex. Despite the neatness of that genetic code, every gene cannot be simply linked to a single outcome. We talked about this in genetics, didn't we? We are not as simple as pea plants. I cannot just link one gene to height. That is a polygenic trait. So some genes are expressed only at certain times or under specific conditions. Like if I jump out and surprise you, or if someone like pulls a knife on you, you release a lot of what immediately? Adrenaline, right? That's a protein, by the way. Okay? So that doesn't happen all the time, or we'd be running around like that squirrel off of over the hedge, you know, hammy or whatever it is, all wired up. So variations and mistakes can occur at each of the steps in replication and expression. Now here's the thing. If my DNA is screwed up, my RNA is screwed up. If my RNA is screwed up, I got the wrong codon, I got the wrong amino acid, and then I changed my polypeptide and made me my protein shape, right? That can be a problem. If I don't code for a stop codon, I might just keep going, right? What if I code for a stop codon too soon? Then I got half a polypeptide that doesn't work. So the final outcome of gene expression is affected by the environment of the cells, the presence of other cells, and the timing of gene expression. When does timing of gene expression really come into play? Everybody say the first 9 months of my life. Right? Think about it. Mom's body telling that little cell, that pile of cells, what proteins to make and what proteins not to make. It's called differentiation. First thing your cells do is they decide if they're going to be a mesoderm, ectoderm, or mesoderm, ecto, or endoderm. Okay? Those are the 3 germ layers of who you are. Basically your skin, your muscles, and your gut. After that, it decides to be what part it's going to be. Now, could any of the cells in your body make all 30,000 proteins? What do you guys think? Everybody say no. No. It's way too much for one factory, right? I have never seen a Nike factory make a GM vehicle, right? So we have different cells that have different genes turned off and different genes turned on. So out of 30,000 genes, I might only make 1,000 of them in like my skin cell. But my liver cell is going to make a different 1,000. My brain cell is going to make a different, right? Because they're all different. That's why I can't just take a skin cell and put it in your heart and have it work. Okay? They're different. And that they're different based on which proteins they make and which ones they don't. So what are stem cells? They're a jobless cell, right? They can become anything. They're like you guys, right? You can be anything. We've all heard that, right? Yeah. You're all stems. Okay. So gene expression, you guys, produces proteins by transcription and translation. Transcription in the nucleus, translation out of the nucleus. This process will take place in 2 stages. Both involve RNA. If I ask you the stage where all 3 RNAs come together, what do you tell me? Translation, because that's where you have rRNA, mRNA, and tRNA all interacting together. And in cells, there's 3 types of RNA. We talked about them. mRNA has the codons. tRNA has the anticodons and the amino acid. And rRNA is the stuff that ribosomes are made of. During transcription, the information in a specific region of DNA is transcribed or copied onto messenger RNA in 3-letter words called codons. Now, the genetic code is based on codons. We use that codon chart to find out which of the 20 amino acids it codes for. Please remember that there are 20 different amino acids, but there are 64 different codons, so some codons will code for the same amino acid. Sometimes you have more than one codon for each amino acid. So translation, we know, happens in steps, and this involves three kinds of RNA: rRNA, tRNA, and mRNA. mRNA codons are read by tRNA anticodons, which carry an amino acid. Two amino acids next to each other form a peptide bond. And the relationship between genes and their effects is complex. Despite the neatness of the genetic code, every gene cannot be linked to just one outcome. Remember, there are polygenic traits. And also remember that your cells are specialized. Stem cells don't have a job. They can be anything. They haven't been told which proteins to make or not to make. But all of your cells at this point, most of them I should say, have been told which ones to make and which ones not to make. That's why your cells are different. Now, I will have, in just a moment, your review